Hello, everybody. Welcome back to Medical Myths, the show about what not to do with your body. And I'm a little fired up today, and you might see Spicy Ben come out at some point today because I went on a huge deep dive about this particular subject. I've been wanting to cover it for a while. And today I'm going to do my best to fit within the usual 1.5 hour live stream because it's going to get a bit dense for a lot of it. And I don't want it to seem too strenuous to go through all of this information, but there is a lot to cover. And this is a subject, of course, you can probably read the title and the description that today we are talking about genetic testing, but not just any genetic testing. We're talking about direct to consumer testing. So a lot of you probably heard of 23andMe or Ancestry.com, or there are numerous tests that you can just purchase and get your DNA sequenced. And this has been a big fad in the community. Um, and it has done some good for people. Like there's a quite a few benefits from having this kind of model, but there's also some concerns that I have. And one that we're going to go in more depth on today that is kind of some pseudoscience and some myths that have become more prominent because of the access to testing and people who really bother me, like physicians. I've seen physicians who have taken advantage of misinformation surrounding this type of testing and use it to sell products or use it to sell other bad ideas. Namely, people that buy into this stuff are also anti vaxxers. They are not in favor of pharmaceuticals in general. We're going to look at some of that rhetoric as well. So, um, lots of pros and cons with this particular topic today. So let's just get right into this thing. So the first page I want to look at is just what are we exactly talking about? So there are multiple ways that you can get genetic testing, and I think I'm gonna zoom in on this a little bit so you can see better. This is just an FAQ from the NIH about genetic testing. And I like here, they've broken it down into a table describing the different ways that you can obtain these. So today we're particularly talking about direct to consumer genetic testing, where there's no healthcare professional involved. The consumer, or I don't really like the word consumer too much, um, but the person purchasing this, they just get to purchase it, they buy it, they do the test, they send it into the company, and then the results are sent back to the person who ordered it. So there's no interpretation of the testing involved at all. There's nobody standing in the way to order this. It is just a customer purchases the product and they get the results of the testing that they do. Um, Another type, of course, is provider mediated genetic testing, where the patient, in this case, will go to their healthcare professional and they will say, "Hey, I'm interested in doing this type of testing." And then the customer or the patient will then purchase a test and then go over it with their healthcare professional. And so this way, the report is given directly to the patient, but they are also allowing access to the healthcare professional to kind of add some interpretation and add some guidance. So that's kind of like a hybrid system where it's patient guided, patient ordered, but healthcare professional guided. And then the last one is what a lot of us are familiar with, where your physician or your healthcare professional that is in charge of your care is going to order this test for you. They are going to have it run. They are going to get the results, and then they are going to go over the results with you and the next steps. So we're just going to go over the first one today. So the tests that market directly to people primarily online, typically with advertisements on Facebook, on Twitter. It is direct to consumer marketing without any healthcare intervention. So they have a little timeline of how we got to this place. Most importantly, the Human Genome Project was completed. So they sequenced the human genome, and people capitalized on this. They said, this is pretty cool. We might be able to individualize medications, might be able to individualize medical treatment by knowing what different genetic variants people have, and further be able to study these genetic variants. And so there were some really good things that came out of the Human Genome Project, but we also got this direct to consumer marketing for genetic testing where people said, hey, you could just know what is in your DNA. And initially it was priced about $1,000, and then they dropped the price down to $99 in 2011, or in 2012. And then it's kind of been that way since, where you could just buy these tests, and they've continued to market them. And so initially starting with a perspective of curiosity, and then kind of leading into, oh, well, we could find out our ancestry. We can find out who we're related to. A lot of religious communities will utilize this, namely the Mormon community will utilize this in their ancestry programs. And then it has evolved to companies saying, well, you don't just have to know about your ancestry, you could know about your healthcare risks. And people who know things about BRCA mutations, about DNA involved in Alzheimer's disease, etc., have had a curiosity in knowing what their risks are. And we'll get into some of those tests and why there's some nuance in who should get these tests and who shouldn't. Um, but then we start getting surfacing of direct to consumer genetic testing for supplement recommendations, for diet recommendations, and it has just gone completely off the deep end to where people can advertise a genetic test for any number of things that doesn't have to have evidence or legitimacy or indications behind any of it. And so that's essentially one of the biggest advantages to a direct to consumer model, is that you don't have to have evidence. You don't have to justify your reason for advertising this. You can just say, hey, well, you could know about this. And people who don't know any better, who don't know kind of what information this is even going to offer, are looking for answers, and they say, sure, this sounds reasonable, and they go for it. And these companies make money. They're not going to make as much money by going through a provider-based model, because there are plenty of tests that people want to run that really aren't going to change management. It's not going to really change your disease risk. And so that's kind of how we got here. And they go over some of the benefits, and I hate that the search bar just crowds everything. But they go over some of the benefits, kind of like I mentioned. Um, an advantage is that you have agency over your health information. You have agency over your own access to care. So you're not gate kept by the healthcare industry. You can say, I want this information. I am going to pay the money and I'm going to get it. And that is very empowering to a lot of people. It gives you a sense of agency. It gives you the ability to have informed consent about knowledge of your own information. So that is an advantage. Um, it is an advantage in that it provides access to genetic testing that, not everybody has access to a medical geneticist. That's very difficult to get into, and they're very overworked. And insurance coverage could be part of that as well. And so if you say, well, I could buy this test, not have to go through all of these appointments to get there, could I have this information sooner? And will this be better in the long run to have this information up front instead of having to go through all of these barriers to get it? And I can definitely see the desire to have more access like that, and the need to have more access like that. Um, a lot of like non-invasive testing is an advantage to some of these. And then the ability to decide what information your healthcare provider gets is an advantage here, to where the results are given to you. You get to decide if you take it to your healthcare provider or not, and that's your decision. So, of course, it's not all that great. there are a lot of limitations. So they go over BRCA in this section, and I think at some point they get into Alzheimer's. But one of the biggest things is that when you don't have somebody there to help interpret what results you're getting, it can be very easy to misunderstand or to have a really high level of anxiety surrounding these results because BRCA1, we know, is a major contributor to breast cancer. And so having that information earlier sounds like a great idea. But something that we need to take into account is that not all, like, the presence of a particular gene does not mean that you're going to have the condition, and not all probability of um, like the gene having a phenotypic correlation, as I mentioned here. Um, you could see that you have a positive result that could cause you a lot of anxiety, even if you might not even have a strong possibility of getting the particular disease. Something like this that is important to think about is Alzheimer's or other conditions that are very multifactorial, because yes, genetics play a part. However, it's not the only thing. And to make people obsessed with a particular genetic variant that may or may not give them a higher likelihood of a condition can cause unnecessary procedures, unnecessary treatments. And so that's not to say that people shouldn't have access to that information, but you need to be cautious with what tests you get. And having more tests isn't necessarily better for everybody. Um, there are plenty of people who have enough risk factors in their family to where they would warrant a genetic test. Like if you have significant family history of particular cancers, or if you have certain symptoms, etc. Those are reasons to get these tests. But then again, you're not doing it just because you want the knowledge. You're doing it because you're further investigating something else. And so we need to think about something called pre-test probability, which I don't have a slide for because I didn't think we were going to go down this route necessarily. But there's a certain probability before you even run a test whether the test is going to be meaningful or not. And so we use that. Most conditions have a particular like scoring system. For example, like with strep testing is incredibly unreliable. And we have certain criteria that you can use to say, should I even run this test? Is this going to make a difference at all? And if they meet a certain amount of this criteria, it's reasonable to say, let's order this test. And if they don't meet any of the criteria, then you can more reasonably say, the probability of it being this condition without any of these like symptoms, without any of these risk factors, makes it much less likely. So we could look into it later if some things change. But um, that's something that a lot of people don't really think about is the probability going into a test like this does impact the statistics of how this data is going to be utilized and what that means for you going forward. So um, the ability to have a geneticist or some other professional that is well equipped to handle this kind of genetic counseling is very, very useful. So if you're going to do one of these tests, having access to somebody who can interpret and somebody who can manage your care afterwards is very, very important. Um, Something that is very interesting as well that people don't consider is that normally when a provider is ordering a particular test, we are accepting responsibility for management after that. So if I think that it's reasonable enough to order a test, I either have to continue to manage that condition or I'm responsible to refer you to somebody who can do the rest of that management. Or if I decide not to manage it, that's on me. I'm responsible for choosing not to manage it. So when you have somebody that purchases their own genetic testing and they get all of these like positives and negatives back for a whole list of genes, and they come into your office and they say, "Tell me what to do with this thing." And you have no idea like what was ordered or why it was ordered. But now you're expected to be responsible for continuing care for something that you didn't initiate. And it can put an extra burden on the system because something that might not have needed testing at all is now taking up resources from specialists and from geneticists that might have other people that kind of need to take priority. And that's not to say that people shouldn't, um, it's not to say that like people shouldn't be able to get into those places if they need them, but it's the risk that we're adding specialist visits for things that don't need a specialist visit because a certain provider was not prepared to end up taking responsibility for a test that they didn't order. And so that's something that we need to take into consideration. Additionally, we have a situation of revealing some like relationship data that you didn't want to know about, and the other person might not want to know about. And now you have that information. And the question of does this violate somebody else's privacy, knowing that your DNA matches? And then something here that I did see in comments before the stream were that some companies have been allowing law enforcement to access the databases for crime investigation. And so there are significant concerns with privacy and what are they doing with your DNA once they have it? What studies are getting access to it? Of course, you have options to opt into certain like consents for academic purposes or for studies, but I know a lot of the companies have now included a um, like some fine prints about this particular issue. But you need to know that if you are giving your health information to somebody else, there are some risks that this could be happening. So just exercise caution. Um, Definitely make sure that what's happening with your information is something that you want to happen with your information. And if that's not a risk that you want to take, then this is probably not really something to pursue on your end. Um, so they kind of go into what the testing is and all of that. And I think we kind of covered where this goes. So with that, we'll move on a little bit from here. I know I wasn't looking at the chat for a minute. So if anybody has questions, um, this is going to get kind of intense. So the next thing I wanted to address is, oh, hey, we got a subscriber, Dennis. Thank you for the sub. Um, we're going to get into a particular myth that has taken over the internet. And this is with regard to the MTHFR, or I jokingly always read it as the motherfucker gene because that's what it looks like. But you get people who find out that a certain gene is related to certain biological processes, and you get blogs like this one, which says, is the MTHFR mutation interrupting your liver and endocrine health? And the font is very small again on this page, so we're going to zoom in. So they say, science has only recently identified a common gene mutation that is a silent driver of many common ailments of which the public is mostly unaware. Many physicians are still not aware or testing for this important enzyme's presence in our body. The MTHFR gene mutation was once thought to affect 3 to 5% of the population. As a result of improved testing methods, we now find that this mutation is currently estimated to impact 30 to 50% of people, depending on ethnicity. This enzyme has a profound influence over everything from hormone regulation to body toxin removal and numerous nutritional deficiencies. So we have this blog. This isn't the only one. And this is from a site called livermedic.com. There are numerous other websites talking about the same issue, and you can find numerous Facebook support groups, one of which I was kind of reading earlier in the day that is run by a physician who has very questionable takes on this particular topic. But this has become a hot button issue in the kind of natural and holistic medicine community. And we'll get into specifically why. But just looking at this, people who aren't familiar with genetics or aren't familiar with biochemistry could easily be persuaded by the fact that this sounds like it makes sense. It sounds like they did their homework and it sounds like they know what they're doing. So, today we're going to dive more into this particular topic because it's so prevalent and it's so easy to get sucked into. But if you know what holes to poke into this, it doesn't make sense. It doesn't there's no evidence that what they're saying is true here. So we can get into their specific claims about this, and then we will kind of debunk them bit by bit. So, um, the MTHFR is an acronym for the enzyme methylene tetrahydrofolate reductase, often referred to as the detox gene. By whom, I want to know they're saying that, because there are multiple genes that are related to detoxification in the human body, and it has a specific function. But to say it is the detox gene is very incorrect. The actual scope of this enzyme is much wider than the term detox gene would suggest. Yeah, there's a lot of nuance with this. Um, it's responsible for breaking down folic acid into a usable form called 5L methyl tetrahydrofolate, and it is coded primarily in two genes, C677T and A1298C. So they are correct with some of this. This enzyme is responsible for processing folate, all types of folate, into a specific form that is utilized as a coenzyme in the homocysteine pathway, which we will get into. So they're correct on this, and there are these two genes are relevant to this. But these two genes they're specifically listing variants, the two genes variants. So these are not the two genes. These are two variants of the MTHFR gene. So they're not correct with that specific thing. And yeah, we'll get into some of this in a little bit. But they claim that when this enzyme can't be produced or is diminished because of this mutation or partial mutation, then lots of important downstream physiology is disrupted. That sounds reasonable because a lot of physiology works that way. And yes, it does relate to methylation, but let's get into specifically what they say. They say the areas negatively impacted include DNA repair, immune function, detoxification, neurotransmitter production, hormone regulation, mood balancing, energy production, and more. So let's just jump straight ahead and see what specific disease processes they think are caused by this. And I'll get into the specific quote here in a second. So they think here, these two genes, when mutated, are associated with the following: heart disease, heart attacks, strokes, blood clots, peripheral neuropathy, anemia, miscarriages, congenital birth defects, and more. And this other variant, they say, relates to depression, fibromyalgia, chronic fatigue syndrome, migraines, irritable bowel syndrome, memory loss, Alzheimer's and dementia, OCD, bipolar disorder, and more. And I'm going to switch over. Oh, no, I'm just going to scroll down here. So more things that they think are related: addictive behaviors, insomnia, schizophrenia, autism, ADHD, behavioral disorders, anxiety. Wait, there's more: diabetes, cancer. They list autism again, spina bifida, which we'll get into that. Lyme disease, which they're referring to chronic Lyme and not acute Lyme disease, because I'm not sure how this gene specifically affects the bacterial infection of Lyme disease. But you can see they've just listed catch-alls for everything, and chemical sensitivity is on this list, and aging associated problems. They're just listing everything they can possibly think of, and there's more, on this list. Um, and they say that 98% of autistic children have these anomalies, and we'll get into that as well. So basically, this gene variant impacts everything, and everybody has this problem. And do you know why they want everybody to have this problem? Well, let's look at this website a bit more. What's this here on the side? Hold on, we'll scroll up. There's a liver cleanse product advertised right on the side here. Is this a website to sell me things? Let me go to a different one that has similar claims. There's another. That's I don't know if that's the one that I wanted. Oh, here we go. No, that's yeah, this is the same website, but we'll get into that one in a second. Did I close the tab? I have so many tabs open, and I apologize to everybody out there. Oh, here we go. Yeah, it is this one. Okay. So we have here on this MTHFR doctors.com. So these people claim to specialize in this particular thing. But look at what they're saying. Who should get tested for this? Over 80% of the population has at least one mutation within the two main alleles of this gene. So they're saying everybody should get tested. Why should you get tested? Because this affects methylation, which may in turn affect many aspects of your health. They don't specify which ones, but this website, looking more into it, they have the same list as the other one. Pretty much anything you can think of. Essentially, you can't prove it doesn't impact it, so therefore we're going to say that it does. So you should get tested. Do you need a prescription? No, you don't need a prescription because you can just buy the test. Where do you buy the test? Where do you get this? Scroll up a little bit. You get it from this website. Genetic reports, genetic tests up here in blue. They're selling you the test. Of course, they want everybody to get the damn test because they're selling it to you. And if you can say that everybody needs the test, everybody has a risk of having all these health problems, so you should buy this test. And this is where one of the cases where direct to consumer genetic testing is really, really sketchy because now lots of people are going to be buying this. Something else that gets very dangerous with this, we're going to go back to the Liver Medic website. This is another page telling people to avoid medicines. Avoid these medicines. They make this condition much worse. So they're telling people to stop taking antacids, telling people to stop taking cholesterol meds, to stop taking nitrous oxide, so not to use it at the dentist, which not everyone's going to use that, but some people need that. Telling people to stop, at least they're saying here for anti-seizure drugs, don't stop taking these without first discussing this with your physician. Yeah, that's the one correct thing that they've said here. But otherwise, no, do not stop taking your anti-seizure drugs because of this mutation. Do not stop taking your birth control because of this. Do not stop taking your antibiotics for this. Don't stop taking your methotrexate, and don't stop taking these meds. Do not stop. If you have diabetes, this is not going to impact as much as your metformin will. And I promise we'll get into the rationale behind what I'm saying. But these claims are out there. They're trying to generalize what people should do. People, they have no idea about their healthcare, what they need. And they're just saying everybody should treat this the same way. And interestingly, say avoid these. You don't want to deplete methyl groups. Avoid all pharmaceutical drugs, just point blank, all of them. Avoid stress. I mean, yeah, fair. Avoid smoking, fair. Acetaldehyde, arsenic, copper you need in your body. But this site says to avoid copper. There's another one from like this perspective that says that you should not stop taking copper. So that's maybe something that they're contesting. And of course, they have they've formulated, I cannot speak. They made a product to help people. So this liver detox supposedly is going to answer all of your problems. Right? All right. So who's going to order this test? Who wants to? Is anybody out there convinced? Yeah, this absolutely is practicing medicine without a license. This is really, really scary. But let's get into why all this is a problem. And I do want to jump back to one of the previous articles because of a quote that they say. I'm going to jump back to this one because they say those with the MTHFR mutation can't process homocysteine and methionine. This results in vitamin deficiency because B12, B6, folate get used up for this process instead. So they make a big claim here that it might be hard for a lot of people to notice. They are claiming that those with the mutation cannot process homocysteine and methionine. So they are making the claim that there is zero production, that there is zero access to this pathway. So they are claiming that this is an all or nothing process. And while there are some biological processes that work like that, this is not one of them. So let's get into the specific pathway. This is where it is going to get a little bit intense. So bear with me, my friends. This will be kind of a crash course in organic chemistry and some biochemistry as well. So I will quickly show kind of the pathway first, and then we'll get more into this. So, and I've tried to include as many of the sources in the description below. So if you want to follow along, feel free. We are going to jump pretty quickly between them. But if at any point you seem interested, they are down there. And this is not making this any bigger. Here we go. Can you all, you can't see this. Okay. There we go. Found it. Okay. So here is the particular pathway. And now I can fine tune that a bit more. Okay. So we have here the homocysteine pathway, and most vitamins that we utilize are coenzymes, meaning they are not the primary enzyme that does the chemical reaction, but they are often necessary to have that reaction happen. So folic acid or folate, they are both the same compound, but they're slightly different. And I'll get into that in one of the next claims that they make. But essentially, this gene, the MTHFR gene, encodes a particular enzyme, 5,10-methylene tetrahydrofolate reductase, which is responsible for converting folic acid or folate, which enters this little pathway here, is converted to a more usable form by the enzyme, and then that is used. So the 5-methyl tetrahydrofolate is used to remove homocysteine, which is a toxic compound if it builds up. It detoxifies, so it does have a detoxification function in converting homocysteine to methionine, and then there's this cycle. So it takes, and it actually utilizes this methionine for synthetic function. So you can make DNA, you can make proteins, you can make other like essentially you can methylate them. So you can modify them for appropriate usage. So usually methylation is inhibitory, but either way, it helps modify things that we're going to use. So it has some very important function in this cycle, but you do need to get rid of homocysteine. And one of the ways, like I just mentioned, converting from methionine, from homocysteine to methionine, or another option is to convert this homocysteine to cystathionine, which is the form primarily for excretion into the urine. So you can get rid of this through the kidneys, or you can convert it to methionine and then utilize it. And if you're going to convert it to cystathionine, you need to have B6 in order to do that as that coenzyme. But in this process here, for the relationship between homocysteine and methionine, the two most common and most important coenzymes are folate or folic acid and vitamin B12. And so if you have a deficiency in folate, and also if you have a deficiency in vitamin B12, you could have problems with this pathway and have an accumulation of problematic homocysteine. And we do know that high levels of homocysteine can cause notably neural tube defects in infants. That's one of the biggest problems. And namely, like some of the biggest two people like to talk about are spina bifida, where the spinal cord actually protrudes through the spinal column and is exposed to the outside, which can be very, very dangerous. Another one is anencephaly, where a baby is born without their head. Basically, they have like their face, but they're missing their brain mostly in the top of their skull. So that's a big problem. We don't want to have things like that happen. So that's why we emphasize having an adequate source of vitamin B12 and adequate sources of folate and or folic acid in your diet is for this particular pathway. But what people are saying, what these MTHFR docs or liver medic, etc., all these people are claiming that with a particular mutation, this enzyme does not work anymore. And if this enzyme doesn't work, you can't convert folic acid or folate to the more usable form, and therefore cannot convert homocysteine to methionine. And there's a couple problems with this particular argument, because we'll get into that. But one of them that I did want to point out briefly is that there are two pathways to get rid of homocysteine, and you build up a large concentration of homocysteine when you exceed the ability of either pathway to handle it. So even if you have a malfunction in one side of this, if the other side is functioning and you're not exceeding the capacity of that one side that's working, you can still get rid of homocysteine and keep it at safe levels. So something that intentionally, and I think it's intentional, that they don't mention this side of the pathway, because it's easier to sell you on, if you don't have this gene working, then you're going to have problems, buy my product. But we do have this other pathway that is available. So that was kind of a lot. Do we have any questions about this particular piece? If you have questions, feel free to put them in the side chat, and I'll try to get to them within a reasonable time frame. So we'll take this from here, and I do want to see if there's one of these papers that could help kind of make this point. I don't know if they mention it on any of these ones. Oh, okay. I need to go to. I think I closed it. I'm gonna reopen one of the pages. So there's a particular lifestyle and diet that people are trying to get people to undergo, um, in addition to buying these other supplements. So this comes down to another line of misinformation. Um, so just a reminder, the hold these myths in your head before we go into kind of the bigger debunk session, because I want to kind of address the two main premises that they're going off of. The first one, which we just covered, is that a variation in the gene will cause complete malfunction and you will not have detoxification of homocysteine. So that is their first premise, their first argument. Their second argument that they're going to make, in this paper, and I say paper, this blog article, surprisingly from a registered dietitian, who has some questionable thoughts. But the other big argument is that if you have a mutation that doesn't allow you to process folic acid, then you shouldn't be consuming folic acid. And how they get around some of the nuance here, there are two kind of well-known forms of folate. There is folate and there is folic acid. And what she says here kind of highlights this difference and where we talk past each other a lot of the time. So since we realized that folate or folic acid is essential for preventing neural tube defects, we've started fortifying cereals, we started fortifying bread products. A lot of processed food has since had a mandate that you fortify it with folic acid. And so these people are saying that you should not be fortifying the food because some people can't process folic acid appropriately. And they draw a distinct difference between folate and folic acid. And they are going to use the naturalistic fallacy, of course, and say that folate is natural. So how do you get folate in the diet? Folate is easily obtained in fruits, green leafy vegetables, legumes, lentils, citrus, asparagus, beef liver, and avocado. And they are correct where you need to eat them raw, which we will get into, so that the folate stays intact. And so they use a statistic that is technically correct, or like a recommendation that is technically correct. They say adult women and men need 400 micrograms of folate per day, while pregnant need 600 micrograms, and breastfeeding will need 500 micrograms. And yes, that is true. That is the recommendation. However, they are drawing a distinction between folate and folic acid. And they say that folate is natural, as you can find it in the diet, and folic acid is synthetic, which is true. It is true, but they are not as different as people think they are, and we'll get into that as well. And there is a functional difference, but folate in its natural form is not better than folic acid. And in fact, it is less good in quite a few different ways. But they're claiming here that you need this amount of folate per day, when actually the recommendation is for folic acid per day. And we will get into that. So they're going to put this label on folic acid, say that it is synthetic. The body is not able to utilize its synthetic form, the synthetic form of the vitamin folate, which partially true. it does need an extra step of processing, but we will get into that. So now that we have kind of these two big arguments, we can address these further. Didn't one of the websites say that 80% of us have this mutation? Yeah, we're going to get into that too. We're going to get into all of that. I'm trying not to kind of spoil the surprises here. I did not mean to show that. Hold on. All right. So let's jump to a better article. Let's go to an organization that I trust a lot more than people actively selling you things. We're going to go to the CDC. All right. So the CDC has an article addressing this gene variant and folic acid. So they have their nice little summary of 3 key points, which we'll go in more detail on in a second. So they say that people with the MTHFR gene variant can process all types of folate, including folic acid. They also say that getting 400 micrograms of folic acid daily can help prevent neural tube defects. And folic acid intake is more important for determining blood folate levels than having a particular gene variant. And so that's a really good summary of what they get into. So these are some points where the CDC and these other people agree. They agree that this gene provides the necessary processing of folate to make DNA, modify proteins, etc. We agree that there are variants of this particular gene. So they say each person has 2 copies of the MTHFR gene. People get one from their mother, one from their father. The possible genotypes. Here's like a little example of a Punnett square situation. And the most common variant is the C677T, which was mentioned in the previous article as well. And they also mention the A1298C variant. So we agree that this is a gene. We agree that it does detox of homocysteine. We agree that it is a necessary function. We agree that there are variants. However, we actually also, something else we agree on is that there's a lot of people with this mutation. And the CDC will agree that gene variants are common and normal. And this is where we start to differ, is what do we do about that now? So the previous article had said that it is such a common mutation. Everybody has distress. Everybody has pathology. You all need a DNA test and my supplements. But actually, these are common and normal, and more people in the United States have 1 or 2 copies of the C677T variant than don't have it. So more people have the mutations than don't. And so what this means is that we really don't need to do anything about it. There are people who have significantly lower levels, and those people will usually have legitimate symptoms and usually will fit into a syndrome of inability to process folate. And for example, if you had such poor function that you gave somebody folate or folic acid, whichever form, and they did not improve, that would suggest, hey, there's a problem with the processing. But if you're claiming that this mutation gives people no function and knocks out this pathway, but then you give them more folate, that doesn't suggest that the pathway is knocked out, because you still have to process the folate in order for that to happen. So the process is still there. It's just a deficiency of folate, which a lot of people do have deficiencies, or at least, especially before we started fortifying food, more people had folate deficiencies. More people had neural tube defects, and there are known neurologic symptoms associated with folate deficiency and with B12 deficiency. So, yes, these are related, but not in the same way that many of these communities will say that it does. And they specifically address the myth about folic acid and folate. So, it says you may have heard that if you have an MTHFR variant, I'm trying so hard not to use the naughty word, however much I want to. But if you have the one of the variants, that you should avoid folic acid and should take other types of folate, such as uh, like one of the ones that's already in the pathway that we mentioned. But this isn't true. So this isn't at all a 100% knockout process. They can still process folate. Their average amount of folate with the mutation is only slightly lower, about 16% lower than in people um, with the wild type, as we usually call it, genotype. So you're only getting about a 16% decrease in function. The people that have significant distress and significant medical need are people with function between 0 and 34%. So that's a lot lower than the average 16% decrease. So, sure, you're going to have a little bit of a slower time processing through this, but unless you are consuming astronomical amounts of folic acid, you're not going to run into problems. And so, with that, the recommendation, even for people with the variants, you should still have the same amount of folic acid daily, the 400 micrograms of folic acid, especially if you have risk of becoming pregnant. So no, you should not. The CDC is saying that you should not adjust your intake because of this. And they specifically say that folic acid, don't know if they say it in this one. No, it's on a different page. I think it's the next, or not. Um, but they specifically use the term folic acid for a reason, and not folate. And this is where we're going to get into some organic chemistry. So hold on, don't leave just because I said that. This will be um, useful. I hope this segment will be useful. So there is a difference within the structures of folic acid and the structure of folate. I'm going to pull up a different paper. All right. So, the difference in structure between folic acid and naturally occurring folates, and they say here, I'm going to try not to read all of this for you, but folates play an essential role in association with cobalamin and pyridoxine vitamins to synthesize, repair, methylate DNA, and also act as a cofactor in numerous biological functions. Mammalian cells cannot synthesize folates de novo, so you have to get this from dietary sources, including natural folates and synthetic folic acid, is the only way to meet daily requirements. And the difference for natural folates, and they're including natural folic acid in this, only 50% of natural folates is bioavailable, and about 85% of natural folic acid is bioavailable when taken with food, because that's how you're getting that. You're getting, if you are getting folates and folic acid from dietary sources, of course you are getting that when you are eating. So you are reducing the amount that you can actually use. So from your dietary folate intake, you're only utilizing about 50% of that, and 85% of your folic acid that you're eating is going to be utilized. But even there, we see a big difference between 50% and 85% between folate and folic acid. But then when you are not eating, if you take a supplement, say in a prenatal vitamin, you're getting 100% bioavailability of that folic acid. So, how is this so different if these are relatively the same molecule? It's because of stability. They are different in how stable they are kind of in their natural state. Um, so, folates are sensitive to heat, oxygen, and ultraviolet light. Um, and folic acid is much more stable. And so, people will say, well, why are we not using folate? Why are we choosing to use folic acid when we know that it's synthetic? Well, just because something is synthetic doesn't mean that it's bad. And we chose to use the more stable molecule because you're going to get more bang for your buck out of it. If you're eating it with food, you're getting 85% of it available versus the 50%. And this is all related to electron activity. And so we're going to talk about redox reactions. And for people who have never taken organic chemistry, this is for anyone that has taken organic chemistry. I know I heard the sigh from the audience about redox reactions. I promise we're going to keep it simple. We're not going to go too far in the redox weeds here. But electron activity does impact how stable a molecule is. And so um, that's where we would agree with some of the previous articles that you would have to intake your folate, if you're intaking it as folate, raw, because it's sensitive to heat, it's sensitive to pH changes because of electron, um, uh, based on valence electrons. So if you remember back to chemistry, um, how we think about at the atomic level, you have the outer circle of electrons, or I guess like the outer orbits of electrons that want to favor having eight. We know that if you have a full 8 electrons in your outer orbit of electrons, it is going to be more stable. And if you have any number different than that, it's going to be less stable and easier to have chemical reactions occur. So this kind of takes acid base chemistry to a little bit different of a level because oftentimes in general chemistry we think of um, there being a transfer of hydrogen, but all of that essentially comes down to transfer of electrons. And so, using a very simple model here of sodium and chloride, we have sodium that we know has 1 electron on the outside, making it very, very susceptible to having that electron stolen from it by something more negative like chlorine. So, chlorine um, has 7 valence electrons. It wants to have 8. And I say want, not that there's like a actual personification, but it just makes this example a bit easier. Chloride is more stable, or chlorine is more stable in its ion form, where it does have 8 electrons in its outer ring. Sodium is going to be more stable, also having 8, but adding on 7 electrons to sodium is much more difficult than giving an electron away to chloride. So that's why we see sodium oftentimes in a positive charge, because you effectively negate that outer orbit of electrons and you leave it with the next ring, which does have 8. And so it is positive. So it is ionized, but it is more stable, less likely to react. That's why elemental sodium is very explosive and salt isn't, because you have these more stable forms. And then, likewise with chlorine, it's much easier to take an electron from sodium to fill out the 8 than to give up 7 and have 8 that way. And so the two ions we have here have a different role in this redox reaction. So reduction, oddly enough, reduction is not giving away the electron. The one that takes the electron is is the one that is reduced, which sounds very counterintuitive. But we say that because it takes on a more negative charge. So the one that's reduced gets a negative charge. The one that is oxidized has a positive charge. So oxidation is losing electrons, reduction is taking electrons. So in this particular example, the sodium is oxidized and the chloride is reduced. Or should I say the chlorine is reduced to chloride. And so this is what happens with folate. So back to this article here, if we kind of go up, we can see it's not super great. I don't like that diagram as much. But essentially what needs to happen to folic acid is the oxidized form. And it is going to be more stable because the folic acid form, like the way the molecule is structured and the way that the electrons are distributed, it is going to be more stable, taking a more positive form as opposed to a more negative form. So the folate we'd kind of refer to as like the reduced form or like the more basic form, or folic acid, of course, is more acidic form. Um, some molecules favor one or the other. In this case, the structure of folic acid favors a more oxidized form. And so while it can exist in either way, um, if you want it to last and you want it to not interact as much with other molecules, you need it to be more in that oxidized state. And so if we synthetically make folic acid, then we can add that to food, know that it's going to last longer, it's going to be less likely to um, denature or become less active with uh, cooking processes, with pH changes. So, like if you have a citrus, you could more reliably intake folic acid along with citrus than you could folate because some of that's going to be um, converted to other things through those chemical reactions. The reason why, or at least one of the reasons why folate is used in the physiologic process is because in the reduced form, it is going to react. In our bodies, we want these things to react because it functions um, like as a detoxifying agent. Like we use it as part of a coenzyme, as part of a chemical reaction to remove homocysteine and make um, other compounds that are safer. So we want it to be more unstable because instability in this case leads to chemical reactions that are necessary for physiologic function. So that was kind of a lot to say that these are both essentially the same molecule, just in different forms that have different purposes, different uses, and neither one is bad. Folic acid is not worse because it's synthetic. It's also actually better in a lot of ways. And like the CDC was specifically saying, folic acid, because it's stable, because it's more bioavailable, because if you are taking 400 micrograms of folic acid, you are more likely getting all of that into your system and using it, as opposed to um, if you take in the same amount of folate, you're not utilizing all of it, so you're not actually reaching that recommended dosage. So that specific word difference is important, and why that's such a hot button topic between these two communities. But I disagree that it has to be folate. In fact, recommendation is to have it be folic acid, but either one is going to be okay. And as long as you get a decent amount of either, then you should be good to go and not have high risks of neural tube defects if you get pregnant. So this particular article goes into a lot of talk about stability of folates. And so they talk about how much is lost in each of these processes. We have, if you have a natural folate, boiling reduces it by 20 to 95%. Steaming, you lose by somewhere from a 0 to 60%. Blanching, 10 to 95%. Like frozen storage could be 0, could be up to 48%. So you get a lot of variability with naturally occurring folate and less variability with folic acid. So that was like a whole lot of ranting to say something I probably could have said in one sentence. But here's kind of the chemical process showing kind of what happens um, in photo decomposition. If you want to read that, you can at some other point. It's not super relevant here. So going back to, we already kind of went to the CDC article. Is there anything else in that I needed to go over? I don't think so. Okay. So moving on. Yeah, and I don't think I specifically read from the CDC, but I want to make it clear. Their recommendation was to not get that you really don't need the testing because it's not going to change what you should eat, should not really change how you live at all unless you have like a super, super severe mutation. But you know what's hilarious? And I was very amused that for once 23andMe and I agree on a particular subject. And so we have a company that does direct to consumer genetic testing, but they have a very different stance on this than some of the other companies that I mentioned. Oh, and by the way, if you go to one of the websites that I showed, whether it be the MTHFR doctors.com, or if you go to livermedic.com, or you go to one of the Facebook support groups, they've essentially decided that any doctor that agrees with them is an expert on MTHFR gene mutations, and anybody who doesn't agree with them is not an expert. So they've just redefined what they consider expertise. And if you agree with them, then you actually did your homework and you know what you're doing. But if you disagree, then you are just wrong because you disagree. And you got a local chiropractor ad. That's hilarious. But 23andMe has, I'm not surprised that they talk about this subject because people apparently ask them a lot. They say some websites and products have made bold claims that common genetic variants in MTHFR or MTHFR genetic mutations can cause a wide array of health conditions ranging from blood clots to cancer to autism and migraines. Their conclusion, despite lots of research and lots of buzz, the existing scientific data doesn't support the vast majority of claims that common MTHFR variants impact human health. That being said, many other genetic variants can actually impact health, which they're correct on. Yeah, there are some severe ones, but they're actually on board with the evidence, and that's surprising coming from a company that profits from direct to consumer DNA tests. Like they sell these tests, and they don't sell MTHFR tests. They even say that here. We currently do not have an MTHFR report. Like they do not sell this test because it's not evidence-based. So they're going to repeat some of the things that we already talked about. And they say over the past 2 decades, scientists have examined associations between MTHFR, C677T, and A1298C variants and more than 600 medical conditions. Despite thousands of scientific publications, the evidence linking this gene to most of these health conditions is inconclusive or conflicting. For example, some studies report an increased risk of heart disease for individuals with two copies of the C677T variant, while other studies report no association with heart disease. And that is something to think about as well. Is people with uh, homozygous variants or heterozygous variants, and a lot of people don't really know the difference between those. But of course, with a lot of genetics, if you have one functional copy of a particular enzyme, then you can make enough enzyme to function well. But even so, even if you have a homozygous variant, you still typically don't have that significant of a decrease in function because it's not an all or nothing pathway. You still have about that 16% decrease on average, and most people don't really need to change anything because of that. But with the claims of heart disease risk, no evidence to really support that. Same thing with cancer, blood clots, um, and they say the exception: women with two copies um, have appear to have a slightly increased risk of having a child with neural tube defect like spina bifida. And so, contrary to the myth that we've been talking about, the recommendation for people with homozygous C677T, the recommendation is to increase your folic acid supplementation, like is to adhere with the guidelines and possibly even increase it to reduce the risk. So it's not that you should decrease your folic acid supplementation. You should increase it or at least keep it the same as everybody else. So if you have a homozygous mutation, it is more imperative that you get your folic acid. And so, yeah, so they say they do not run this test. And they say we will continue to monitor research in this area as we do for a wide variety of genetic research and evaluate new information as it becomes available. And they say here's kind of the disclaimer. Some rare variants in MTHFR can cause a severe condition called homocysteinuria, which is a result of that huge buildup in um, homocysteine, where if you have a complete malfunction of the methionine pathway, this is just directing all of it into the urinary tract, and that can be problematic um, in fewer than 1 in 200,000 individuals. So there are people that can have severe limitations of this pathway, but that is an extreme exception. And so these pretty large Facebook groups saying that anybody with this mutation is at risk, that's not necessarily true. And it is definitely overhyping something very benign. But if you didn't know kind of these nuances, and if you didn't know things about like phenotypic penetrance, which is a funny way to describe how much of a particular phenotype are you going to get if you have certain genes? Because even if you have a gene that doesn't, and even if it's dominant, doesn't mean that you're going to express it. And that can be very variable depending on what gene we're talking about. And so if you don't know that, you could buy into this, you could get this unnecessary test, and you could think that you are at risk for any number of conditions just because people told you something that was inaccurate and was an over reaction um, to misinformation. And they took some information that was correct. And this is something that pseudoscience loves to do. They will take pieces, little bits and pieces of accurate information and miscommunicate them or misattribute certain conditions to that particular thing when it wasn't at all evidence-based. Um, I do want to jump over to another. I know there's so many articles today, so many. But here's another one from the AHA ASA journal. Um, so this is about homocysteine and the same gene mutation that we've been talking about. They have a more simplified diagram here, but I primarily wanted to look at their explanation of the risks with homocysteinemia. So, okay, so they address what are the risks for someone with elevated homocysteine. So let's say you actually had elevated homocysteine levels. Um, like there are some risks of cardiovascular disease. However, data show that risk is only mildly increased. Um, blood clots, like they can occur, but risk is only slightly increased. And although elevated homocysteine is a risk factor for a first episode of DVT or PE, it does not predict a higher risk of recurrent clot once a patient is off blood thinners. So pretty similar risks and risk management to the general population. And this is for somebody with elevated homocysteine, not just everybody with the gene mutation. This is if you actually have excess homocysteine, like if you actually have problems in the pathway from the gene variant. And then this statement here. So talking about pregnancy complications, preeclampsia, placental abruption, and pregnancy loss. However, it appears that elevated homocysteine levels may be a consequence of these complications rather than a cause. So possibly these things caused an elevation in homocysteine and not the other way around. And their relation to neural tube defects, which we've kind of gone into. It's been in here, and they say it's been investigated as a risk factor for several other conditions, including autism, cognitive impairment, osteoporosis, Down syndrome, migraines, MS. So a bunch of the things that were on the list for the other blogs and websites. However, there hasn't really been any evidence thus far that suggests that, and it's very hard to like use some of the statistics that have been quoted because they'll say that 98% of autistic children that they studied like had this particular mutation. However, if we already have knowledge that the majority of the general population has this mutation, then we can't really make that determination. So they're again relying on a lot of correlation versus causation arguments. And here we go. Can homocysteine levels be lowered? If so, how? Yes, it can be through folic acid supplementation, both using folate and synthetic folic acid. So, opposite of what people are saying we should do. So then they go into a lot of the same thing. But here we go. Here's another statement. Who should be tested for MTHFR mutations? There's no indication for MTHFR mutation testing in routine clinical practice in any patient group. So even if somebody has blood clots or recurrent pregnancy loss, this particular gene is not something to really look into. Kind of the case looking into neural tube defects could be more of a justification. However, you can more easily test this, like with if you gave somebody supplementation, does it improve? And if they respond well to supplementation, then it's probably not as much of a problem. So lots of resources agreeing with the exact same thing. And here's another older paper, now that we're just giving lots and lots of sources today. So here's a little abstract for a paper discussing the updated estimates of neural tube defects prevented by mandatory folic acid fortification. So this is folic acid, the synthetic form, not the natural folate that people want us to have. And this is from the years 1995 to 2011. And here you go. Where's there? Try not to read the whole thing. I'll just read it. Okay. In 1992, the US Public Health Service recommended that all women capable of becoming pregnant consume 400 micrograms of folic acid daily to prevent neural tube defects. NTDs are major birth defects of the brain and spine that occur early in pregnancy as a result of improper closure of the embryonic neural tube, which can lead to death or varying degrees of disability. The two most common neural tube defects are anencephaly and spina bifida. Beginning in 1998, the United States mandated fortification of enriched cereal grain products with 140 micrograms of folic acid per 100 grams. Immediately after mandatory fortification, the birth prevalence of neural tube defect cases declined. Fortification was estimated to avert approximately 1,000 neural tube defect affected pregnancies annually. To provide updated estimates of the birth prevalence of neural tube defects in the period after introduction of mandatory folic acid fortification, data from 19 population-based birth defects surveillance programs in the United States covering the years 1999 through 2011 were examined. After the initial decrease, neural tube defect birth prevalence during post-fortification period has remained relatively stable. The number of births occurring annually without neural tube defects that would otherwise have been affected is approximately 1,326, and that is at a 95% confidence interval. So essentially what we get to with this confidence interval, the number here, 1,326, falls within this 95% confidence interval. So it's not enough to reject the null, essentially. So this is saying that, so mandatory folic acid fortification remains an effective public health intervention. So what we were doing, we fortified the grain products with folic acid. We evaluated the rates of neural tube defects, and then we did it again later on to see if this number had changed throughout this whole time that we left the standard where it was for fortification. And so what we saw was that it was relatively unchanged throughout that, and there was not enough evidence to reject the thing that we'd already been doing. So this essentially did not change what we had known beforehand. So I think I had another source I wanted to go over. My other thought would be, do we go into one of the public groups on Facebook to see some more of those claims? Because that can be kind of funny. We'll do it for a few minutes. We'll do it. So let me go over to Facebook. Alrighty. Just make sure I have everything safe for viewing. So this is a public group, so I'm not sharing any private information. These are things shared to a public page that anybody can access. So this is a support group for the MTHFR gene mutation. And this is run by a physician, unfortunately, with some very interesting takes. Lots of people post things about their supplements, whatever. So Carol Savage is the physician who makes a lot of very generalized statements. Like there's a section where she goes on about potassium, which that's a whole other topic, but like I agree with the general premise that we want to keep our sodium within normal range. Too high and too low is not good. However, she posts that and then posts, where is it? She posts EKGs, sample EKGs of people with potassium derangements, or just like examples of potassium derangements. Where? Oh, it's pretty far back. I guess they were posting a lot today. Which, oh, real quick before we find that, I guess somebody asked, "Is the tetanus vaccine toxic for us?" And so, and then of course somebody responds, "It is to my children, but of course they won't label it as an adverse reaction." So we have some anti-vax things shared in this group as well that aren't being moderated out. So that's an acceptable viewpoint to have in this group, which tells us a lot. But like for the potassium, yeah. So I agree that we want to have appropriate ranges of potassium. And then she posts like these EKGs of hypokalemia, which like, yeah, she does mention that this is too little potassium, less than 2.5 milliequivalents per liter, which is incredibly low. We are talking a level where you are destabilizing the cardiac membrane and can get arrhythmias. Here's the problem when we start talking like this to lay people who think that anything outside of the reference range or anything like on the lower end of the reference range is going to cause this, which it's not. Like we're talking a very significantly low level and very significant symptoms. We're not talking just like generalized like fatigue, like not feeling your best. Like people hear low potassium and anything that's been low for them, they will equate to being an emergency like this, and they will wonder, like looking at this, they will say, this probably explains my fatigue. It felt like adrenal fatigue, which is not a thing. And they drink an adrenal cocktail each day to help with that. So there's clearly a disconnect in how we're communicating these types of issues with lay people. And so even if a physician posts accurate information like that, this represents less than 2.5 milliequivalents per liter, people don't know what that means. So if you make a post like this and say that, like suggest that all these symptoms are related to low potassium, really anybody is going to read that and think, oh, I have low potassium, my heart's going to give out and I'm going to die. Probably not. Like probably not. And so that's the rhetoric that they're going off of with these gene mutations, and they go over a few different genes that they will obsess about. But here's kind of a point that she made that was similar to the all or nothing rhetoric of one of the other articles, saying that those with the MTHFR C677T variant are likely lacking in the ability to make enough of the reduced folate called L-methylfolate that donates its methyl group to naked B12 to make methyl B12. Then methyl B12 is supposed to donate that methyl group onto homocysteine to turn homocysteine into methionine. When this biochemistry doesn't happen as it should because of this gene variant, then homocysteine becomes elevated, again suggesting this all or nothing pathway, which, as we mentioned, is BS. That's not how this works. And it's not that like all of your copies are malfunctional, and it's not that you can't do anything with it. It is just less effective and like less efficient. Um, so communicating that this is completely non-functional or that it is non-functional enough to completely derail the whole pathway is dishonest in the communication. And then this person, of course, asks for ways to lower homocysteine, and they recommend this book and go into things like a whole rant and how you should not consume folic acid. Yeah, and then here, if you already know that you have, and this is the doctor again, if you already know that you have the variants, folic acid should be avoided, which we know is false. And then this person says, no reason to take a pharmaceutical. Your doctor knows nothing about MTHFR if she's okay with folic acid. There's a lot of good quality supplements that work for you. So anybody that disagrees is bad. And none of them give data for these claims. And that's something too. When we looked at the other websites, there are no citations except for their own products. Same thing with the Liver Medic. You have this whole page of like medications to avoid. I don't see a single citation. I don't see a single citation on this page. So it's literally just them making claims, and that's it. Yeah, it felt like adrenal fatigue is a great diagnostic method. Yeah, like, and this is why I'm not a fan of self-diagnosis. There are a lot of ways, like, as at the end of the day, patients absolutely, they know their bodies, they know what they feel, they know their experiences, and this isn't to discount any of that. But clinical reasoning goes beyond saying this disease process that I looked up on the internet matches my symptoms, because there's so much overlap. Like, we think about chronic fatigue. Like, let's think of a differential for things that cause like chronic fatigue. Sure, you could have a vitamin B deficiency, it could do that. You could have sleep apnea that can do that. You could have depression that can do that. You could have autoimmune diseases that can do that. Malnutrition, uh, like medication reactions, like allergies, if they're severe enough, asthma, like if you're not breathing well, really any pulmonary conditions can cause fatigue. Heart failure can cause fatigue, like pericarditis can cause it, like really any number of cardiovascular conditions. Hypothyroidism can cause fatigue. Like, I have a huge list of differentials that if I just went to the internet and said, I have this thing, and someone gave me an answer of it's obviously this gene mutation, and I get the test and it says, yes, you do have the gene mutation. The problem with that is if I've already made that self-diagnosis, even with a genetic test, like even if I have objective data that says I have some contributing factor, what I'm going to think of then is I'm going to probably anchor on the thing that I read and say it's got to be this thing because it matches. And then I stop doing my work and I stop getting evaluated and I stop looking for a better answer. And then people can get worse. Like, there are consequences to self-diagnosis, and there's a reason why we have to be very careful about thorough investigation and not just giving into whatever the supposed answer in front of us is. And I feel for these people. I feel for people that have unanswered medical questions, and unexplained symptoms. But it's better for symptoms to be unexplained and continually investigated than to have a wrong answer. And so I think that ultimately, these tests, I don't think that they're inherently bad. I don't think that if you wanted answers about your genetic sequencing or about your any genetic mutations or variants you might have, I don't think inherently that's bad. But we need to have realistic expectations about what we're getting out of this, what we expect to do with it, if anything. Like if it's just, I want to know about my body, I want to have the information, and I don't really care about anything beyond that, cool. But know that that would be a reason you're doing it. If you're doing it to pursue additional treatment or additional evaluation, it's important to have a discussion with somebody who can interpret the results with you and can talk with you about what your options are after that. Like if I get this test for the MTHFR mutation, what's going to change? Is it worth me spending $100 or more than $100 to get this answer? And for some people, the answer is yes, it's worth it because they want to know. And that's okay. But you have to understand that there might not be anything we can do after that. And the most likely answer might be just keep doing what you're doing. Or if you want some of these answers and you want to pursue further treatment, it's important to have someone that can give you that. And let's say, you had something that came up and you talked with your provider and they said, here's your probabilities. Would you like an evaluation by a geneticist? And you can then go down those pathways to get more evaluation. But again, risking, like, is it worth potential unnecessary surgeries, unnecessary lab tests, unnecessary office visits, unnecessary costs for office visits to have that done? Like, is this particular mutation worth pursuing that for you? And if yes, then maybe it's worth doing. But if not, maybe it's time to take a step back and be okay with sticking with the guidelines and being okay with not knowing. Yeah, it definitely. I always think about it as that terminology. I've been trying to use more PG language today, because I know some people don't like some of that extra language, but I do think about that every single time. And Dennis had a question about my opinion on British osteopaths. That's a question I could answer a different day. But the long and short of it, osteopathy in other countries is viewed differently as it is in the US. As far as I know, DOs in the UK are not trained to be physicians. So I would say no. I would say they're on par with chiropractors in the way that they're trained in the UK. However, in the US, this is different. And yeah, we got the chiropractor ad, which is hilarious. But I think I've covered everything that I want to cover for today. Thank you everybody for sticking with me on kind of a more nitty-gritty sciencey topic. If anybody has questions about redox reactions or about the folate processing pathway, anything that we covered today, feel free to leave comments or questions in the comment section. And even if you want to bring things up in future live streams, totally okay with that. There are communities on Facebook and Discord where you can talk about these topics as well. I try to be available to answer questions. Sometimes I'm not as good about that. I'm going to try to be better. So if you want to ask questions in the Discord or Facebook, that's totally okay as well. I did not have trivia yesterday, but hoping to get back on that relatively soon. I think that's all the announcements that I have for this week. I'm not sure what next week's topic is going to be, but stay tuned. I appreciate having you all here, and I'll see you all in the next one. Bye, everyone.